recent discovery in patients with lupus.
We've been trying to understand the regulation of this autoimmune T-cell response, essentially the flavor of the immune response that gets activated in patients with lupus.
Could result in better therapeutic treatments.
The experiments that we did led to a new idea about a therapeutic strategy to treat lupus.
And later, learn about research focused on lupus diagnostic errors.
For some people, it is a very long circuitous path, and for others, the diagnosis happened more quickly. So there is so much heterogeneity in the disease, but also in getting a diagnosis.
Discover a research breakthrough and diagnostic challenges of lupus inside this edition of CTSI Discovery Radio. Welcome to CTSI Discovery Radio. I'm your host, Brian Belmer. CTSI Discovery Radio is brought to you by the Clinical and Translational Science Institute of Southeast Wisconsin. The CTSI is a consortium of researchers, doctors, scientists, and others representing 8 institutions, including the Medical College of Wisconsin, Milwaukee School of Engineering, Marquette University, the University of Wisconsin-Milwaukee, Children's Wisconsin, Froedtert Hospital, Versity, Blood Center of Wisconsin, and the Zablocki VA Medical Center. The CTSI works collaboratively across all of our member institutions. Our mission is advancing health through research and discovery. Lupus is an autoimmune disease that can cause pain and suffering for people with it. We've talked about lupus previously. Check out episode number 70 of our show. But today, we first focus on a research breakthrough it's hoped leads to improved treatments for patients with lupus. For this, we spoke with Dr. Deepak Rao, Associate Professor, Department of Medicine, Division of Medical Sciences at Harvard Medical School, and Principal Investigator of the Rao Lab at Brigham and Women's Hospital in Boston. For review, Dr. Rao gives a brief description of what systemic lupus erythematosus, or SLE, is.
Systemic lupus erythematosus is a broad autoimmune disease where the immune system mounts a response against our own proteins and components. The disease can show up in a number of different ways. It can cause a wide range of symptoms, oftentimes rashes, joint pain, fatigue. Sometimes it can attack organs like the kidneys, causing a condition of lupus nephritis or injury to the kidneys.
He adds, this autoimmune disease can take many forms,
which becomes a challenge for both patients and physicians who are trying to diagnose it. But at its heart, it's an autoimmune response that attacks different parts of the body.
However, recent study findings were published on the discovery of a molecular defect leading to SLE.
We know that lupus involves an autoimmune response where T cells and B cells that are reactive to the self initiate an immune response that then causes this damage in multiple places. We've been trying to understand the regulation of this autoimmune T-cell response in patients with lupus, essentially the flavor of the immune response that gets activated in patients with lupus,
with the specific goal of then
being able to redirect that immune response or suppress that immune response to dampen the effects of the disease. We've known already that both autoreactive T cells and B cells become activated in patients with lupus. We know that this is a central part of the disease. So our project was about trying to understand the types of T cells that get activated that are contributing to this immune response, and then trying to identify factors that are critical for the functions of those T cells that we could manipulate to try and slow down the response.
In the study, lupus patients shared distinct molecular changes, causing an imbalance in the types of T cells they generated.
T cells are a type of immune cell whose job it is to recognize when there's some foreign component around, usually a foreign protein. It's circulating around the body, and it's looking for some foreign protein that it can see with its T-cell receptor. And when it sees something foreign, it becomes activated to send a signal to other types of immune cells and other cells around that there's a problem.
The T cells are differentiated into subset types with different functions.
One class of T cells called T helper cells, and a separate class of killer T cells or cytotoxic T cells. Now, amongst this helper T cells, there are many different types. Helper T cells can be divided into subsets that have specific jobs. The ones that we think are particularly relevant in patients with lupus are a set of T helper cells particularly effective at interacting with B cells and helping to stimulate B cells to make antibodies, which is the job of a B cell.
An imbalance between the helper and harmful T cells can significantly impact a person's health.
As we looked at the composition of the T cells that are present in blood of patients with lupus have too many B cell helper T cells. We think that it's this excess. There's too many of them. They're interacting with B cells in an abnormal or excessive way, and it's stimulating an abnormal, excessive B cell response and the production of autoantibodies. And that's really a core feature that we see in patients with lupus.
Dr. Rao tells us this type of autoimmune response,
T cells interacting with B cells, this is a prominent feature of lupus and one that we think is a key driver. In patients with lupus, what we noticed was that not only do they have too many B cell helper T cells, but at the same time, they have too few of a different subset of helper T cells called Th22 cells. Their roles are a little bit unclear, but one of the things that they do is to help maintain the integrity of our barriers, like the barrier in your guts, the barrier in your skin.
and the pattern that emerged in patients with lupus
is that on one hand, they have too many of these B cell helper T cells, which stimulate the B cell response. And on the other hand, too few of these Th22 cells that we associate with barrier protection. And it's that imbalance in the T cell population that seems to be a typical feature that we see in patients with lupus.
This discovery was made by comparing the blood of lupus patients to a control group.
We started with a pretty simple initial effort, which is to collect blood samples from patients with lupus who are interested to participate in the study, as well as controls who don't have an autoimmune condition but are similar in age and sex. And in both cases, people who are willing to participate donate a vial or two of blood. We isolate the immune cells out of that blood, and then we profile the immune cells to ask about the composition of the different types of immune cells in the mix. We're essentially quantifying different types of T cells or B cells or other types of immune cells, and asking about whether any particular types of T cells are overrepresented or underrepresented in the samples that come from patients with lupus compared to control.
What makes this study particularly interesting is that it started with human models.
This is really a human immunology study. There are no animal models in this project. This is a project that started with making straightforward observations about what happens in the immune system in patients with lupus versus controls, and then using that as the starting point to come up with ideas about the patterns that we see, things that are different in a patient with lupus versus controls, and then testing some of those ideas in vitro using cells from people, taking advantage of these new technologies that exist.
What kinds of new technologies?
Things like CRISPR, which allows us to disrupt the expression of different proteins in cells from people, using these broad, powerful genomic studies to measure broad patterns of gene expression or the binding of specific proteins throughout the genome, where they go and how they do it.
A study using this type of human immunology approach
can yield not only new insights about the disease, but also new fundamental concepts about how components of the immune system are regulated. In this case, T cells specialize to help B cells. The idea that those two cell populations are regulated in a coordinated way, this wasn't known. And this came not from animal models, but from observations coming from patients, and then bypasses the question about whether the work that you're doing can be translated to humans. We start by doing it in humans.
In addition to the T-cell imbalance, Dr. Rao's research identified a protein called interferon.
Interferon is a factor that was recognized decades ago as a signal that cells will produce, especially in the presence of a viral infection. And cells will make it to signal to other cells around that there is something wrong and that all the other cells around should be on high alert and should make themselves less susceptible to viral infection. The original name interferon was exactly this idea, something that cells would make that would interfere with the ability of a virus to infect other cells.
While it's been known for decades that
patients with lupus make far too much interferon, it's one of the characteristic features of this autoimmune disease. patients with lupus have this overproduction of interferon. When you look at cell types you can collect from a patient with lupus, they all share this signature of their response to interferon, changing the environment in total.
What's newly discovered from this research study?
One of the things that interferon is doing in patients with lupus is it's pushing T cells towards an acquisition of this B cell helper function. And we think this is part of what the damaging role of interferon is in patients with lupus, is to stimulate this abnormal set of T cell B cell interactions that enhance the production of autoantibodies.
His research further discovered activation of a protein preventing T cells from developing into lupus-causing cells.
This, I think, is one of the most interesting parts of the study. We did this with my close collaborator and friend, Jay Choi, who's at Northwestern, to try and ask which specific signaling molecules within cells are important for their ability to turn a otherwise undifferentiated T cell into a B cell helper T cell.
It turns out that
one striking factor that came out of this was a protein called the aryl hydrocarbon receptor. It's labeled as a receptor, but it's actually a protein that regulates the expression of many genes.
this aryl hydrocarbon receptor, or AHR protein, does a couple of things.
It binds to DNA and will turn on the expression of specific genes. And it turns out that this action of the aryl hydrocarbon receptor does a couple of things in T cells. For one, it pushes T cells to become a Th22 cell. That's already been known. But what came out of this study was that another important thing that the aryl hydrocarbon receptor is doing is it's preventing T cells from becoming B cell helper T cells. The action of the aryl hydrocarbon receptor is suppressing the generation of B cell helper T cells.
So now, knowing this,
when we disrupt the gene that encodes the aryl hydrocarbon receptor so that cells can't make it, those cells are much more likely to turn into B cell helper T cells. And in a number of experiments where we can either activate or inhibit the activity of the aryl hydrocarbon receptor using small molecules that function as activators or inhibitors, you can push T cells one direction or the other by either activating or inhibiting AHR.
Together, it's hoped that these discoveries reveal an improved strategy for treating lupus.
The experiments that we did and the identification of the aryl hydrocarbon receptor as the receptor that can control the differentiation of these B cell helper T cells led to a new idea about a therapeutic strategy to treat lupus.
The idea is,
if we can activate the aryl hydrocarbon receptor in T cells, we should be able to suppress their differentiation into B cell helper T cells and slow down this pathologic T cell B cell interaction that goes on in patients with lupus.
But there are challenges.
To activate the aryl hydrocarbon receptor in T cells without activating it in all cells, because essentially all cells have the aryl hydrocarbon receptor expressed, and many different types of cells will respond to activators of AHR in many different ways that can lead to toxicities or side effects.
So instead, the idea is to
find ways where we can selectively activate the aryl hydrocarbon receptor in T cells to prevent their differentiation into B cell helper T cells, which we think fuel this autoimmune response in lupus, and instead move them towards other T cell phenotypes that may be benign or even protective, for example, Th22 cells.
Leading to treatment for lupus that is hopefully more targeted and more effective.
Using a strategy that selectively targets T cell differentiation towards a particular pathway, we can suppress what we think is the pathologic response in lupus without disrupting the function of T cells more broadly or without disrupting the function of other immune cells that are important for defending against infections.
While this sounds promising, it's early in the process.
We don't actually know what's going to happen here as we try this. There's a lot of unknowns here, but the idea we think is an exciting one.
Could this new therapy strategy be useful for all lupus patients?
There's a sense that this disease is very heterogeneous. It causes a range of different manifestations and symptoms. At the same time, there are core features consistent amongst the majority of patients with lupus. And I think a therapy that's targeting this pathway should be relevant across the majority of patients. It's one that's going to make sense for most patients who have lupus.
So if you, or someone you know, are battling the effects of lupus,
there are a set of physicians and scientists who care very much about trying to do better in terms of ways that we can reduce the burden of this disease on patients. This kind of research depends on the participation of both patients and physicians who are really dedicated to these kinds of studies, and we really appreciate and depend on the participation of the whole community.
Next, we discover a qualitative research study involving rheumatologists to explore diagnosis challenges of lupus, including delayed and misdiagnosis of the disease. We spoke with Dr. Julia Simard, Associate Professor in the Department of Epidemiology and Population Health and in the Department of Medicine, Divisions of Immunology, Rheumatology, and Obstetrics at Stanford University School of Medicine. Dr. Simard begins by explaining what rheumatology is and what a rheumatologist does.
Rheumatology is a medical specialty that focuses on diagnosing and managing musculoskeletal and connective tissue diseases. So this includes diseases like rheumatoid arthritis, systemic lupus, systemic sclerosis, and it's particularly relevant to our discussion today because many lupus patients are cared for and diagnosed by rheumatologists.
Many diseases can be challenging to diagnose. How common are diagnosis errors, such as misdiagnosis or delayed diagnosis?
That's a really interesting question and one that I think a lot about, but I don't think we actually have an answer to this. Methodologically, this is really hard to answer reliably because we don't have a way that we track that necessarily. So unfortunately, this is a really interesting area of research and really important, but I don't think we have a way that we can reliably quantify that at this point.
But Dr. Simard recognizes some specific consequences of diagnostic errors.
So, for example, if it's something that requires treatment to avoid really bad outcomes, progressive disease, then these types of errors can be a big problem because the longer it takes to get treatment, the more likely these bad outcomes are, right? So if a disease is progressive, you might see irreversible damage. If the disease may lead to death, then you're increasing people's chances of bad outcomes. And so it really does depend on what disease or condition you're talking about.
In the case of something more serious, like a malignant cancer,
if a cancer is missed, then it likely progresses, right? So you go across stages and then maybe you have metastasis.
compared to something with less serious consequences.
It could mean people feel unwell for longer than they need to. It can lead to inability to work, loss of quality of life, loss of function, and that maybe once they start treatment, they're feeling back to kind of normal. So the consequences of these types of diagnostic errors or just diagnostic delays vary by disease, but can be inconvenient delays, just feeling unwell, to having really bad consequences.
What about with systemic lupus erythematosus, or SLE specifically?
It's really hard to quantify how many diagnoses are missed. We actually don't have a number that we can assign to this, but the consequences will really vary. Lupus is a very heterogeneous disease. Lupus doesn't necessarily look the same in all patients.
And so, whereas some lupus patients might experience
fevers, joint pain, they might have certain rashes, certain immunologic abnormalities like autoantibodies.
Others with the same diagnosis might experience more severe symptoms.
Brain involvement, they might have seizures or other issues. They may have kidney involvement and have kidney disease as a consequence. It really does depend on what type of lupus somebody may have.
Either way, a delay in diagnosis
means that someone is not getting treated. Not being treated means they are likely to progress. We don't have a way to yet effectively study this at the population level, but if you talk to patients, for some people, it is a very long, circuitous path, and for others, the diagnosis happened more quickly. So there is so much heterogeneity in the disease, but also in getting a diagnosis.
Are there specific factors contributing to diagnosis errors for the disease?
Yeah, it's a really good question. We conducted a randomized survey study. Basically, we created patient summaries summaries. A insert gender, insert race patient that is X number of years old comes in with these complaints. This is their family history. Here are their clinical findings. Here's what you find on exam. Here are their labs.
Physicians were sent a survey with vignettes of two different hypothetical patients.
One that was kind of a mild to moderate SLE, and another that was this phenotype of lupus nephritis, which is lupus that affects the kidneys. And we also added a kidney biopsy.
For the physicians receiving the survey,
we randomized whether the version of the hypothetical patient they saw was black or white, and then a woman or a man. In the literature, it's very common to see lupus described as more common in black or African American women in the U.S. compared to a white man. We were interested to see if we randomized demographics of the case and held everything else constant, did that change the diagnosis that rheumatologists assigned to that case?
What they found is for the mild to moderate lupus case vignette, physicians surveyed were
much more likely to get it right when it was presented as a black woman versus when it was presented as a white man. And then we further wanted to know, is this for all lupus or is this for mild to moderate SLE that hasn't really affected any organs yet? And so we had another version where it was lupus nephritis and there were clear biopsy findings as well as lab results.
And when the disease was this more severe phenotype,
we did not see as big of differences when we looked at the race-gender combinations, although we did still see that they did a little bit worse when the case was presented as a white man.
She adds that
when a man is diagnosed with lupus, he tends to have more severe features like face rash or kidney disease. We kept finding the group that represents the non-classical version of the disease seemed to have either more obvious features of the disease at diagnosis or had more severe versions or phenotypes of the disease.
So, this randomized survey experiment got Dr. Simard and her research team thinking.
What's going on? We've held everything constant. We held their age constant, their family history, the lab findings are identical, and yet we still thought, well, what's going on? Is this suggesting some sort of cognitive biases or cognitive errors?
Going into the randomized survey experiment, there was some concern over possible biases.
When somebody is not the classical case that is maybe taught in medical school, it is harder to see the possibility of this diagnosis.
So in the case of, say, a white male,
if we say this is a disease that predominantly affects women of color in the U.S., then when a white man comes in with the same symptoms, just by default, lupus is just lower on that differential list. But then once all the lab results start coming in, we need to think about, with all of these symptoms, maybe I need to move this up to the top of the list.
The findings of this survey resulted in a follow-up qualitative study.
We interviewed rheumatologists to understand and contextualize the findings from our randomized survey experiment. Because we didn't really know what was going on, we had some of our own hypotheses, but we wanted to speak to rheumatologists for them to tell us, does this make sense? And if so, why do you think this is happening? And what are the consequences to the patients?
Dr. Simard shares some specific details about that study.
The study included 41 rheumatologists. 2/3 were women. The majority had more than 10 years experience since completing their fellowship. Just under 20% reported being 5 to 10 years out of their fellowship. And the interviews were an average of about 45 minutes.
The researchers also spent time
getting acquainted, learning a little bit more about their practice, how commonly they see lupus patients, how they were trained. And then we actually presented the results from our randomized survey study to them.
After sharing these results, the rheumatologists were asked a series of questions.
We asked them, why are we seeing these results? We then talked about what maybe were the consequences to patients. This bias is really playing out. Really important to remember that this experiment used hypothetical case vignettes that aren't real people, so it's very possible it doesn't 100% represent how they would practice in person with a live patient in front of them.
Then some more questions.
Because we were also interested in whether there was an underdiagnosis of lupus in men, we then also asked some general questions about the diagnosis of lupus in men, how common it was, how it presents in men, if it's different from women. And then we wrapped it up thinking about, is there anything we can do to combat these potential biases?
5 main themes were identified from the interviews.
The first focused on how the training they've received to approach the diagnostic process and clinical decision-making thinks about archetypes, thinks about race and sex, and how common something is. And so that's related to this theme, relying almost on the epidemiology of lupus being more common in Black women versus white men.
The next 2 themes involved
using sociodemographic cues to trigger almost like pattern recognition that developed as a test-taking strategy, which is then also related to the 4th theme, which was using these patient vignettes might trigger people to think back to test-taking strategies. Would we see the same thing with hypothetical patients, right? Just more of a question of, are these results the consequence of it being a survey rather than live people in front of them?
And the 5th theme identified
this idea of varied consequences to patients related to potential bias here. There were a few sort of sub-themes that came out of it. Delayed or misdiagnosed could lead to more progressed outcomes. The increase in cost, potential increases in harm and suffering, just a worse prognosis for long-term outcomes. Does this lead to loss of trust in clinicians and the medical system?
What conclusions were reached with this qualitative study? Did it help identify challenges of diagnosing lupus?
The thematic analysis of 41 interviews with rheumatologists did identify several potential causes or contributors to misdiagnosis, missed diagnosis, and delayed diagnosis in lupus among men, including this idea of reinforcing of sociodemographic characteristics as cognitive shortcuts in medical examination and decision-making. We also found that potentially the use of race or sex-based archetypes for lupus diagnosis are present in clinical training that might be contributing, and this sort of reliance on the epidemiology of lupus as a disease most commonly seen in women, and particularly women of color, may be contributing. Those were really our main conclusions,
which were related, at least somewhat, to what was anticipated.
And so now we've put this work out there and are speaking with clinicians, with groups of rheumatologists to sort of learn more and discuss how do we move forward.
In the meantime, the study has already provided some benefit.
One of the biggest benefits of this work has been engaging rheumatologists and clinicians in this discussion, thinking about how cognitive biases may be contributing to some delays in care, some misdiagnosis, and having them be open to thinking through whether there might be changes that could be implemented in training.
And that's only the beginning.
I feel like this is the start of a conversation, and people seem very receptive to having this conversation, which is really exciting.
Still, there's more to come.
This is going to be a longer discussion around quantifying the extent to which there are diagnostic errors. It's really hard to know how big of a problem this is, but we know that consequences of it can be quite bad for some people because of the complexity. And so there is really an interest in the community to figure out how can we improve this.
But now, that's all the time we have for this edition of CTSI Discovery Radio. As always, thanks to each of our guests for appearing on today's show, Dr. Deepak Rao and Dr. Julia Simard. I hope you've discovered something by listening to today's show, and I'm doubly hopeful you'll join us again next time. CTSI Discovery Radio airs the third Friday of every month. Make an appointment on your calendar and join us for each episode. On behalf of the Clinical and Translational Science Institute of Southeast Wisconsin and all of our affiliate partners and members, I'm Brian Belmer, wishing you happier, healthier days ahead. For more information about research or to listen to the podcast of this or any of our shows on demand, please visit our website at ctsi.mcw.edu. CTSI Discovery Radio is written, produced, and hosted by Brian Belmer in collaboration with WMSE Radio. The CTSI and this program are under the direction of Dr. Reza Shakir.